just after 6, so I think we'll go ahead and get going. I want to introduce to you my graduate student, Jesse Humbert, that is going to be presenting his thesis research tonight. So, this is Jesse, and I imagine I first met him at some point during a class I taught for him, like, for instance, Intro to Biological Research or Cell Biology or something like that. Probably Intro to Biological Research. I say imagine because I was actually trying to think about the first time I met Jesse today, and I actually have no memory of him in those 2 classes at least. Which is really kind of weird to me because if you know much of Jesse, he's usually the kind of person that makes an impression, like a pretty stark impression. My first actual memories of him distinctly as an individual human comes from Rosario several years after I imagine I taught him in those classes when him and another student took on a project in Dave Cowles' class looking at octopuses, which, you know, that caught my attention finally. That whole story up until now probably says a lot more about me and my attention than it does about Jesse. But after that point, Jesse definitely did make quite the impression. Sometime after that, he began working with me on a research project along with Brian Hartman to put together ocean acidification project materials for the high school classroom. And then after that, he ended up working with me as a master's student. Now, this is a picture that I took at Rosario during spring of last year. This is probably about this time last year, when he is kind of in the thick of trying to figure out his research project. Now, I picked this one because it actually encompasses several different things I want to explain about Jesse. I think this is kind of like a good picture to encompass several different things. Let me talk about one of them. Number one, he's wearing a mask. This is actually kind of interesting because his entire graduate program has been during the pandemic, and this has actually been kind of a big issue. His first year, the last year, I taught no on-campus classes that entire year. It was shortened. We didn't have nearly as much time to work together. Yet everything that he accomplished despite that, which I found pretty impressive. Let's talk about other things. He's holding the camera system, which you're going to hear more about tonight, which is highly modified. This thing right here that you're seeing is not this thing that you see right here, which talks to a little bit of his resourcefulness. He found something that he was interested in pursuing and modified it to his own purposes, which I think is impressive. There's another thing. There was a dog. So he had a dog out at Rosario, but this kind of illustrates another thing about him. This dog recently had puppies. And how many puppies did it have? 11 puppies. And you're selling them for how much a piece? Yeah, like $2,000 a piece. Like there is a strong entrepreneurial streak in Jesse that I've never had in a graduate student before, and it's served him well. Also, the dog is carrying the rope. I feel like there's meaning in that, but I can't really connect it to anything particular at the moment. And just in general, this kind of captures a lot of Jesse's persona here. I mean, going along with that kind of independent entrepreneurial streak, like he showed up at Rosario at the beginning of spring and informed me that he had bought a sailboat. Cool. Randomly. And then had a lot of good times on it last summer and then sold it at the end of the summer. So we had a boat for the summer, which, I mean, just it felt like it also just kind of encapsulated a little bit of who Jesse was or is. I don't mean to speak to like you just died. I'm sorry. Anyhow, so I've really enjoyed having Jesse in the lab. He's brought a very unique energy and spirit to the lab that I've never had before and probably won't have again. He is a singular person, but he has been a great addition to the lab. So with that, I will give the time over to him to let him tell you about the research that he did.
Wow, thank you. That was very emotional. I appreciate the kind words, and hopefully you guys enjoy my thesis presentation. So Does the mic look good and everything? Okay, awesome. So my name is Jesse Humbert, as you've previously mentioned, and I'm doing my work on Octopus Rubescence. So I've completed my thesis through kind of the thick of COVID, but hopefully gain a little bit of understanding for Octopus Rubescence and their social behavior. So let's start by examining the subject of my research, Octopus Rubescence. This is called the ruby octopus. It's common throughout Puget Sound. You find it from Alaska all the way down to kind of Baja, California. They're very common around Puget Sound, although they are pretty difficult to find. This is mostly due because they're just so small. They can kind of squeeze through, fit through anything. They're cryptic. They can change their color, their texture, kind of their shape. So when you get underwater, it often sees you long before you see it, and it's gone. It's blended in with whatever it's next to. It's under it. It's beside it. It's squeezed through it somehow. They're just gone. So very little work has been done with the social behavior of this octopus. Part of that can be attributed to the fact that they're just hard to find. The work that has been done is faced with these limitations. So a lot of these studies have taken wild octopus. They've put them in kind of captive environments, aquariums, things like that, and then watch their social behavior. These limitations change the behavior of an octopus. If you take something that's normally pretty solitary and you cram it in a tank with a bunch of its conspecifics, its neighbors, it's not really going to enjoy that much. Also, if you remove factors, resources, and limit those, it's going to be more aggressive as well. So by constraining their range and their resources, you can often take an animal that's not normally very aggressive and kind of make it aggressive. So we've also long anticipated that Octopus rubescens is nocturnal, and this is based largely off of what we've observed in aquariums in captivity, not off of wild studies. And the reason for this is people are walking through aquariums all day long. If you're going to be nocturnal or diurnal, you're not going to be out during the day if people are banging on your tank and walking around and looking at you and things like that. So this is largely based on that, and although it hasn't been proven yet, my anticipation is that the reason that they're mostly nocturnal and we have this kind of concept of that is because people are just bothering them all day long in captivity, and so why be active during the day when you have all night to just be alone with your thoughts? So let's jump over to the questions I want to try and address in my master's. So for my thesis, I wanted to better understand the denning behavior of Octopus rubescens. So dens are super important for octopus. They spend the majority of their lives tucked away in these dens. They rely on their dens for protection from predators, to safely eat prey, reproduction, etc. So octopus dens are found throughout Puget Sound, and they can be anything from under a log, in an old tire, a trash can. There's even some toilets in our dive site that are occupied by octopus. Basically anything that's kind of rigid and strong, they'll find a way to inhabit it. They're very, very resourceful. In this area, however, the one that I'm going to be talking about today, it's primarily composed of glass bottles that have kind of fallen down this large embankment and scattered across the sea floor. So almost the entire kind of area that we dive is just covered in these glass bottles, and it's very, very easy to find octopus on a regular basis. So basically, octopus and their dens, if we just focus on those, they indicate such a large portion of octopus life history that you can get a really good estimation of their entire life, how they operate, their behavior, conspecific, interspecific interactions, foraging behavior, how often they come and go, when they bring prey. I mean, the list goes on. But they spend 80% of their life in their bottles and around that area, so it's very easy to survey one area and capture a huge portion of that octopus's life history. So, with that context, I focus on 3 different questions. First off, what is their general daily activity? So what do they do on a day-to-day basis? How often do they go out hunting? When do they bring back food? Mating, interspecies relationships, all kinds of things. Then I focus on how do they interact with interspecifics. So interspecifics is any animal, any organism that isn't that species. So this can be a fish, a crab, it can be a different species of octopus. How do they interact? And then finally, how do they interact with conspecifics? And conspecifics are other octopus in the same species. So basically, their friends, their compatriots, how do they interact on a day-to-day basis? So first, let's look at where we can even find this many octopus, let alone capture them. And then let's come back to these 3 questions. So as I previously mentioned, near our marine lab in Puget Sound, we discovered an area full of octopus living almost exclusively in bottles. And this presents a really unique opportunity. All of a sudden, it's really easy to find a lot of octopus, and it's very easy to collect them. The population is rather easy to gauge and monitor. How many are there? What are they doing? It's easy to collect a good majority of the population. This is really important if you want to try and get a kind of a concept of the population as a whole. If you catch one or two octopus from a variety of different areas, it's not really going to tell you what the entire population is doing. I've attached a short video here of an octopus capture, and this should give you a good idea of what it looks like. Shout out to Brian Watkins, who I think is watching. He videoed this for me. So in this video, an octopus is running along the sea floor. This is kind of rare. Usually they're inside bottles. We throw them in these Ziploc bags, and then we pop them into a mesh collection bag. There you can hear Brian offering some commentary. So we tuck it in these Ziploc bags, we throw it in a mesh bag, and when we get to the surface, we pop it out, put them in an aerated saltwater cooler, and transport them back to the marine lab. So now that I have a good spot to capture a lot of octopus, here comes the next problem. How do I tell an octopus apart and observe their behavior? As I just mentioned, octopus can change their color, their texture, their shape, their size. It makes it very tricky to tell one individual from another, especially at a distance. If you had enough time to just look at one octopus, and you could weigh it, and you could do things like that, it'd be pretty easy to tell which one is which, if you could sex them, things like that. But from a distance, it's very hard to tell them apart, especially if they're trying to camouflage. So to solve this issue, people have tried to tag octopuses with a variety of different tags. They use chemical, electronic, external, internal, tattooing, branding, radio tags. I mean, the list goes on. It's pretty crazy, honestly. Most of these are used for much larger octopus. The giant Pacific octopus, which can get several hundred pounds and can handle a large tag punched through its arm. These guys, however, are so much smaller, under a pound, that we have to kind of improvise our technique. So all of these techniques that I've mentioned have often been met with pretty high mortality rates. Not many survive, and very few individuals even recover from it. So in order to remain minimally invasive, because I can't disturb this entire population, especially if I want to make assumptions about how they interact with one another, what they do on a day-to-day basis, if I punch a giant hole through its arm and put on a big radio tag, it's not going to behave normally. Okay? So to combat this, I use this relatively newer tech called ViaTagging, and this is used more commonly for salmon and fish tracking, things like that. Whoa, here we go. Will that start on its own? There we go. Okay. So ViaTagging stands for Visible Implant Elastomer. And this is basically like a biocompatible elastomer that you inject as a pliable solid, and then it kind of like hardens. It will sit below clear or translucent tissue, and it fluoresces at certain wavelengths. So normally it's not very obvious, and as we get into some of these photos, you can tell this is a little more obvious here, just kind of in daylight. But if it's under the skin and there's any kind of a texture or coloration to it, it's very hard to distinguish under the skin. So hopefully it won't impact kind of predatory influence or anything like that. Additionally, we never had any necrosis, like skin damage, anything like that from these tags. And some of the longest retaining tags were like 6 months. We're seeing them on cameras 6 months later, doing fine, just trucking along. So ideally, these have a very, very low impact, not like previous tagging methods. So now that I have a way to distinguish one octopus from another, as you can tell, all of these kind of have an individual tagging pattern, and they're all custom, and then all of these are photographed and logged with their sex, their weight, when they were collected, when they were released, all that kind of information. So I've essentially found a way to isolate each octopus and make it an individual for my eyes only if I use a fluorescent light. So now let's jump over to how would I even monitor octopus, especially wild octopus denning behavior. Even though I can tell them apart, how would I continue with this? And I'm sure many of you are thinking like, oh, that's super easy. You just take a GoPro, stick it on like a stake, stick it underwater. like put it on an octopus den, just leave it there, right? Put it on a time-lapse mode, like that makes a lot of sense. There are a few intricacies to this that make that a little bit more difficult than you would anticipate. So first off, GoPros, they wouldn't be able to distinguish an octopus from another because I need a like fluorescent light to be able to do that. Secondly, an octopus coming and going is a relatively rare event. It might happen 4 or 5 times a day at best. If you have a GoPro set up at a 10-second interval, the odds of you missing an octopus crossing like a 2 square foot area is very, very high. So either coming and going from the bottle you would miss, there's too much error in that. So a small GoPro camera like that as well doesn't have much battery life or storage space, so it wouldn't be able to run very long, and it would require a lighting setup to run throughout the night. So in order to run a time lapse every 10 seconds, the average GoPro would only last a few hours, and that's not good enough. We need it to run for days, ideally. So really just putting a time-lapse camera, any kind of a time-lapse camera, above their dens won't really work. We need something that would work a little bit better than this. So the system I designed is basically an underwater motion detecting camera, and it has kind of a few unique abilities which allow me to observe octopus and tell them each apart. So let's look at the construction kind of piece by piece, and then we'll kind of come back to these questions we've stated in the beginning. So the foundation of the computer component relies on a Raspberry Pi 3B+, and this is just kind of a consumer-based, open source like computer system. You can program it to run all kinds of different electronics. So this board on top is called a PCB, and it's basically a custom circuit board that allows us to run different components. So this runs like a clock, it runs a strobe system, runs a little LCD screen so we can see what's going on, what the voltage is, things like that. So let's look at the computer component. So for this, this is an ArduCam with a CSV wide-angle lens on the top. I'll try not to get like into all the nitty-gritty stuff that's not super important, but basically these camera bases are 20, 30 bucks. You can just buy them online. You attach them with a ribbon cable into the actual computer that I just showed you, and they are sensitive to all the visible spectrum. They have a filter, as do most cameras, that prevent them from seeing IR and far red. So most animals, as you know, game cameras like that work off IR. This won't work for us because underwater, IR, basically heat, dissipates so fast that you're not able to actually, it doesn't accept it back into the camera. So any kind of a lighting system utilizing IR or sensors utilizing IR would not work, especially for an animal that is the same temperature as the water around it. So to make these cameras sensitive to those wavelengths, I removed those filters. So as we look at some of the photos, you'll be like, why is it kind of pink or red? That's because it's removed, the filters are removed. So now we're looking at the lighting in the camera system all kind of built together. So the Raspberry Pi that runs our camera system, and then the custom PCB which fires our strobes. So that strobe attachment connects to kind of a whole strobe housing, and that provides all of our lights underwater. So let's jump over and look at the LEDs. So this system utilizes 3 high-powered LEDs. So 2 of the LEDs are up on top there. They're blue, and those are used for fluorescing the tattoos. So those are UV LEDs. Then the bottom one is a far red LED, which octopus aren't able to see. So it's basically kind of like night vision for the camera. It fluoresces, it shines, it'll blink every second, and that'll illuminate the entire area day or night. So if you get a cloud going over, even though there would be a dip normally during the day, this will still illuminate the area so you can see everything that's going on. So this is mounted in this kind of housing system that slides into these ports. So there's like these ports. This is the system, in case you're wondering kind of what it looks like. The LEDs and the camera all slide into those ports and mount in there so they can see out and see what's going on. So let's jump over and look at the structural components. So the camera system, all the structural components were fabricated from 3D printed parts. So the batteries are housed in these kind of like black encasing, the battery mounts, and there are 3 batteries that are placed in this. Then in the very base here is the computer component of it, which is mounted in that kind of lower frame. All of these are 3D fabricated, and then they're housed using these threaded metal rods. They're secured in place using nuts. So the nuts, when they're torqued down, it actually makes this entire system super rigid. So if I were to pull it out of this entire housing, I could hold it just by this, and there would be no lagging in the system. It would be like nice and rigid, which is very useful when you're trying to load it and unload it from a big PVC housing. So let's jump over and look at the batteries really quick. So to power the system, battery packs were made by wiring essentially 2 Roomba batteries together. So these are robotic vacuum batteries. It's what we needed because it has a very high amperage and a very low voltage. Because we don't need a lot of voltage to run this thing. 12 volts run just about every electronic you need. Low voltage with high amperage. So we would take 2 batteries, tether them together, wire them in parallel, and then we take 3 of those batteries and we mount them together to make 3 battery packs. When wired together in parallel, it produced an average of about 27,000 milliamp hours, which seems very impressive, but it only runs the system for about 3 days. Granted, this is because it's strobing every second, and the LED strobes, all of those take a lot of energy, as well as the computer, which is using algorithms to detect if there's motion. So the 3 battery packs are all wired together. This wiring harness kind of tucks into these side components, these clips on the sides of the battery mounts, to keep it all contained so it doesn't get like cut up or tangled up inside the housing. So let's jump over and look at the housing. The entire system is mounted in a Schedule 80 PVC housing. If you don't know much about PVC, which I didn't, Schedule 80 is the highest rating kind of PVC that you can get, and it's based for high pressure applications. So if you want to put a ton of pressure through these pipes, you get this stuff. It's about $50 per fitting for these. So this is about $3 worth of PVC, which is kind of exorbitant, but that's based on the pressure rating for it. So this housing can actually go down to about 800 feet, even though it's just made out of like plastic pipe. The mounting on the ends of it, so these are the kind of port windows. This port here has a high pressure switch. So this is made by Blue Robotics. It can go down about 3,000 feet. It's meant for deep sea drones and stuff like that. Things that you can turn on when they're underwater. So this is a waterproof switch that allows us, after we set up the camera system, to actually turn it on before we leave, as opposed to trying to set it on timers and things like that, and getting it perfectly set up, and then the strobing starts. So we can turn it on when we're ready. And these 2 ports over here, one of those is for our LEDs and one is for our camera system. Jump over and look at like a cutaway view. So this is our entire housing made out of PVC with our camera system sitting inside. So it's the exact same system you're seeing here, but it's just flipped. So we have our camera, which tethers into our Raspberry Pi, and our system tethers into our strobing system as well. So plexiglass covers that go over each port allow the camera and the strobe to shine out and us to be able to see what our LCD screen is reading. Is it on? Is it voltage looking good? What's happening? So now we'll jump over and look at the deployment frames. So these frames I welded to house the camera systems. They're made out of angle iron, flat iron, and then they're just welded together and then coated with marine paint to prevent any rust. So every about 71 hours, when a camera was done with the deployment, I'd have to go out, dive the site, remove this camera system, and replace it with a fully charged empty one. And by empty, I mean it doesn't have any photos on it. It's ready to accept a bunch more data. So I designed the deployment frames with a hinge door, and this allows me to easily exchange cameras. It takes me about 30 seconds, maybe a minute, to swap a camera, and then we're on to the next deployment frame to do the next camera. So let's jump over now and look at just kind of a few photos of what that looks like. There we go. So here's just a few images from deployments. You see the end of the camera housing here. You can see inside the LCD screen and the electronics, hopefully still dry. Here looking up underneath the camera system, I'm checking to make sure that the port windows are clear, that we don't have any leakage, anything like that. And then on the far 2 left images, the green one there, I'm taking a camera out and attaching it onto my BCD so I can take it up with me. The difficulty with these guys are they're about 30 pounds each, and so even just having 2 or 3 on your BCD is like about 90 pounds worth of weight that you're trying to lug around while you're swimming. And it makes it kind of difficult. It was a lot of good training, so that was beneficial. The far left, that's just kind of a mock-up of what it looks like an octopus sitting outside of a bottle right below the deployment frame. And that's kind of your ideal deployment scenario. Now let's jump over and look at data collection. So now that I've explained how all the components kind of fit together and how it all works, let's look at what we found from this. This is by far the most interesting part of this. The cameras ran for about 33 days in total, and we got a few different types of photos collected during the deployments. So this is based on kind of its motion detecting feature. So on the far left, we have our background photos, and it takes about 25 photos and layers these together. I can set it for more if there's a lot going on. And this is basically helps the computer detect whenever motion changes. So this background is then compared with every evaluation photo, which is taken every second, and it compares these together to see if motion is changing. So right before this trigger photo is taken, an octopus's arm kind of encroached into that evaluation picture, it triggered, and then it switches from a low resolution black and white photo to a high quality color, far red, and UV photo. So the trick with this is if you take a photo every second, you don't want to come back and evaluate that. So this is basically like a GoPro on steroids in that we're doing kind of like a time lapse every second. And then whenever we detect motion, we take an actual kind of a good photo. So these are the ones that we primarily analyzed, but we can jump back and look at those evaluation photos. For instance, a fish swims by really fast. We get maybe 2 shots of that. I can jump back and I can see this in a second by second frame as opposed to just like one photo of it, okay? Additionally, I want you to note that in the high quality color photos, the UV is strobing as well, and this is so we can fluoresce our tattoos, but there's no reason to have UV strobing when there's no motion detected. Okay, let's jump over and look at how we analyze all this data, how we kind of organize it so we can do statistical analysis. So this is an event log, and I mean, there's like thousands of these entries now on a giant Excel spreadsheet, and I want to give a big shout out to my undergraduates who did a lot of this work for me. But they came through and basically gave us an octo ID, what octopus was it, when was it found. We synchronize all that data with the octopus that we found back into the water. Which bottle is it occupying? Is it a non-resident? Is it a resident? I'll kind of address these terms in a minute. What species is it? And then what behavior is it performing? So we can have non-Octopus Rubescence, like a cancer crab, that's coming and going and being preyed upon and doing all kinds of different behaviors. Okay, let's jump over now and look at some terminology. So I gave a mock-up here of kind of what's going on. This is pretty simple, so bear with me, but we have a resident octopus. That's basically the octopus that's occupying the bottle beneath the camera and staying kind of within that camera frame area. Okay. We have a non-resident octopus. This is one that is not occupying the bottle or the frame, and it's encroaching or passing through the area. So the second it enters a bottle, it then switches over to becoming a resident octopus, and it's occupying the site. Okay? The camera frame is basically the area that the camera can capture beneath it. Anything beyond that does not concern us, so we use that as kind of our sample area. Anytime we have a frame departure or a frame arrival, that is an octopus or any species coming and going from that camera frame. So, although we evaluated over about 20 different behaviors, I'm going to address 2 of them, fortification and periscoping behaviors, and then we'll look at some figures in a minute that kind of tie into that. So fortification, this is basically any time we have an octopus covering the den entrance with material. So this can be shells, rocks, other bottles with octopus in them. Sometimes they'll drag an entire bottle over their entrance, large rocks, things like that. So we start off at time 0, and this is basically an octopus was introduced to the bottle. It is occupying the bottle. After about 2 hours, we see the amount of debris it's pulled onto the top of its bottle has changed significantly, and by 4 hours, it's grabbed this giant rock and pulled it over the entrance. So this behavior is called fortification behavior, and we'll get to kind of like when do they do this most frequently, why do they do it in a minute. Periscoping behavior, This is pretty self-explanatory as well, but this is anytime an octopus keeps the majority of its body inside its bottle for protection, but sticks its eyes out so it can see what's going on. So this is a pretty good example of it. You see the octopus's eyes here, and he's holding like a shell there. This was his little door that he pushed away so he could peek out, and then if something spooks him, which in this deployment, every crab, every little fish, anything that came by would scare him. He jumps back in the bottle, puts his little shell up, and a few minutes later he comes back out and starts periscoping again. So that's the behavior we're looking at here. So now I'm going to jump over and look at this video, and this is basically like a time-lapse field every time motion was triggered. Just playing basically like a time-lapse video. So there's two bottles on the bottom here. This one here and this one as well. Both of those are occupied with a resident octopus. They've been living here for a while, and we're going to watch kind of what happens on a day-to-day basis for an octopus. So here's another. This is the third Octopus Rubescence in the frame. He's coming up. He's trying to reach inside this bottle. He's not having like great luck with it because there's already an octopus living inside that bottle. So he's starting to kind of leave the camera frame here. This other bottle over here has a lot of fortification material in it. I just showed you this bottle over a 4-hour period. That was that fortification event I was demonstrating. Now we have a new, smaller Octopus Rubescence come in. He's doing the exact same thing, reaching in each bottle kind of as he goes past. He likes this bottle, so he starts kind of down on top of the octopus, which is already living in that bottle. So he's kind of packed him down below all of his fortification material. You're probably noticing now that this octopus that just started inhabiting, this non-resident, has this orange marking on its head. Those are the fluorescent dyes that are showing up. He has some other markings, but it's a little hard to see. There's a blue and a yellow stripe further down his mantle. So he's kind of grappling with his neighbor here. There's a good shot of the orange. And then because he's not able to get all the way down in the bottle, he dramatically exits it and starts to leave the camera frame. So the next shot here, you'll see his tentacles as he jets away. Sorry, his arms. And the reason for this quick departure was a shark coming in right behind him. He wasn't able to get down in the bottle because of all those fortification materials, so he had to quickly leave the frame. So the shark is pursuing him. I will have to look at more deployment to see if we actually found him again or if he got eaten possibly there. But you see the shark continue to circle in this area. Here's a large rockfish coming by. Another rockfish. And there's the shark kind of nosing around, smelling the bottles, trying to figure out what's kind of going on. And I'm obviously personifying this a lot more than it is, but it makes the story a little more interesting. So now as the shark is leaving, we see another rockfish sitting right below the frame. They like to sit right below the frame. Often they go up on side and you just get a huge shot of an eyeball. You're going along late at night looking at these photos and then boom, there's this huge eyeball. Always scares me. So this video is a good example of denning behavior. This is just like the other one. There's a lot of activity in a pretty short amount of time. So this guy right now is periscoping, the small rubescence up on the top. And you'll notice, kind of as he changes his colors, you'll start to see those markings. You'll see fish coming by, the currents, like little things disturbing the soil. There's a lot going on. So he goes out on a little forage, walks around for a little bit, comes back. And as it starts to get towards the evening, you'll notice that it's becoming more red. And this is just because there's less influence from the sunlight. So as evening approaches, he's starting to inhabit this bottle. He's now switched. There's a big rockfish comes by. And you see those markings are becoming more and more obvious, those bright yellow markings at the top of his mantle. So he continues to inhabit this bottle. He's kind of cleaning it. He's kind of moving things around. It seems to be just kind of surveying the area. Then here in a minute, we'll see a much larger octopus move in. Oh, crab scared him off. It's a red rock crab moving through. Now the tags are really, really visible. And here about now, you'll see a large octopus suddenly move in and cover his bottle entrance. And this larger octopus, I'm not really sure what it's doing, if it's trying to reach inside the bottle, but it will remain there for a while. There's a small fish going by. There we go. So now we have a much larger octopus, and he's not trying to evict. We assume he's just kind of sitting here and holding the bottle. So he remains there for a while, and then he moves into the adjacent bottle up on top. Same species, yes. Much larger one, though. So now, through the rest of the night, he fortifies. He keeps reaching his tentacle over and into his neighbor's bottle just to kind of annoy him, and he passes the night having a great time, fortifying and annoying his neighbor. So let's look at a few other photos of different species that we've captured. So these are, we got some wolf eels, we got a painted greenling, we got some flatfish down here as well. And this is just to kind of accentuate the use of this for any kind of benthic ecology. You want to survey fish or crabs, worms that live in the dirt, whatever you want. This can survey the ground, the benthic areas, very well. Here's some lyncods. We never observe these feeding on octopus, but they have a very impressive set of teeth. They're pretty aggressive and they're quite large. And a lot of people have found octopus in their stomach contents. So it's kind of strange that they didn't actually prey on any of the Octopus Rubescence, despite having pretty ample opportunities. Then here's a few images of what things look like at night. So it's nice and red here, as you'll see. We have a chimera here. We have a rockfish as well, a small octopus with a sea star. And it's a little hard to tell here, but there's a shark's head cruising by at night. So now that we've kind of seen what the data look like, let's look at our results. So as I said before, I had a team of 3 undergraduate students working to categorize all these behaviors, record the times, the species, the interactions, all kinds of things. We collected roughly 250,000 trigger photos. These are the colored ones. And we got about 3 to 4 million of the evaluation photos. So it took a long time to analyze them, but here are some of the results. So this is focusing on frame departures. Anytime an octopus left the frame, and we kind of denote this as like it's out and about. We don't want to say that it's foraging, but it's out interacting with the outside environment beyond that one camera frame. So this is a radial histogram. Imagine it as like a 24-hour clock. So we have midnight up here. We have noon down here. The blue is denoted by the night period, so you have sunset and sunrise, and then this yellow is our daylight. So it's a pretty easy graph to read, but all of these orange areas are basically periods whenever we saw this behavior. So every time we saw a departure, it would add to one of these orange periods. So we broke it into hour categories, we looked at the timestamps, and this is what we found. So it seems pretty obvious probably, but we found a big preference for being active during the day and very little activity during the night. So this is in pretty stark contrast to what I mentioned before, where we largely think these animals are nocturnal. So this stands in pretty direct defiance of that. Additionally, we have our N values. So our N represents basically how many samples did we find, how often did this occur. So we got 47 of those, and then our P value indicates that this is statistically significant.
Good bankers, 9 to 5.
Yeah. We'll jump over and look at fortification now. So we found some interesting thing in regards to den fortification. So a fortification event, like I said before, is anytime you pull stuff into the den entrance to kind of protect yourself or just block it off. And we found that octopus almost exclusively fortify their dens at night. And this, when compared to the previous graph, they're out active during the day, nighttime is approaching, they're departing less and less, they're kind of petering off. They find a bottle that is appropriate for the night, and then they start fortifying the bottle. So the first 2 hours after sunset, they're fortifying, they're pulling in shells, debris, they're trying to presumably protect themselves from larger predators, or maybe even conspecifics, other octopus trying to invade their bottle. And they're able to fortify for a few hours, and it kind of peters off as the night progresses. And then sunrise comes, they're out and about, foraging, roaming around, doing whatever they want. So now that we've looked at kind of this general behavior, we know there are kind of day-to-day cycles. Let's jump over and look at interspecific interactions. And these are basically anytime an octopus interacts with a species other than its own. So here is a very big graph on interspecific interactions. So on our y-axis over here, we have visitation duration. Visitation duration is basically how long did this organism hang out next to these octopus bottles within the camera frame. Then we have visitation frequency, how often did they come to the camera frame. And then on the very bottom, our x-axis, we have whether or not an octopus is present. So we have absent, present, all across the bottom for each species. So visitation duration, how long did the species stay here when octopus was absent versus how long was it there when an octopus was present. And then how many times did it come when it was present versus absent. This bottom portion here is based on basically how often would we expect it to occur if it was reliant on just like pure average. So if it just happened randomly and we didn't have any preference for one or the other, we would see in like an ideal world like a 50-50 split. That's not necessarily what we see here, but that 50-50 split would represent it's just as likely to occur when an octopus is present versus when it is absent. Okay? So if this is blue and a positive number, it means it's more likely to occur than we would expect. If it is red and negative, it means it's less likely to occur than we would expect, based on this kind of 50-50 ideal scenario distribution. So as we go across the bottom, you can see which ones are more likely and less likely to occur when an octopus is present or absent. Up here, we just have like a standard box plot. So I'll start with the species and kind of go across the top. We start off with Hexagrammus decagrammus, and this is a kelp. So these are fairly large fish. They're kind of known to prey on octopus on occasion, but in this instance, we found kind of an interesting relationship that isn't based on any kind of previous knowledge on these species. So their statistical value, it's both significant for the duration that they spend with octopus and how often they come by. So you'll see that they come by more frequently when octopus are present, and they spend about 3 times longer when octopus are present. So they go out of their way to come when an octopus is there, and they spend much more time. Ophidium elongatus, which is just your lincod. These guys, in contrast to the kelp greenling, don't come by more frequently. They don't spend more time. They just kind of do their thing. Sebastis, these are all their rockfish species. We kind of clumped them all together because that's a little easier than looking at 5 or 6 different rockfish species. They were very significant. However, they have a tendency to avoid coming by when an octopus is present. So they're actively avoiding these areas when an octopus is there. And they come by significantly more when the octopuses are gone. We see the same trend for Cancer productus, which are red rock crabs, and that makes a lot of sense because if you're a prey species of an octopus, any kind of an octopus smell or an octopus vibe, you probably wouldn't want to hang out there for any long duration. So we actually see that they don't spend more or less time there when an octopus is there. They do avoid coming by when octopuses are denning. So these two species actually avoid it, whereas this one on both sides, duration and frequency, target octopus. Now on the other side, this is more conspecific than interspecific, but we're looking at non-resident Octopus rubescens. So this is basically an octopus that's visiting the area, but not actually living in those bottles. So we find that they actually spend, and it's tricky here because these look very similar, but they spend a significantly longer amount of time when an octopus is present versus when it's absent. And the reason this is significant, even though it looks very close, is because this is based on a logarithmic scale, and so it's exponentially larger than what you see down here. We just had to break this axis because it is a very large table. So if I highlight these to kind of describe what's a positive interaction, what's a negative interaction, basically these 3 prefer interacting with octopus. I don't want to say target them, but they kind of go out of their way to target octopus. And then the red areas down here, they avoid octopus. Okay? And this is generalized a little bit, but it should make a little bit of sense. Now that we've kind of seen this kelp greenling interaction, this one's unique. The frequency and the duration are greater when an octopus is present. Let's jump over and look at a little bit more in depth on these kelp greenling interactions. So these images are taken at a 5-second interval. This is as fast as the camera can do all the kind of complex coding, if you will, to determine if motion is detected, take a evaluation photo, and then capture an image. So these are taken at like a 5-second increment. And you see this octopus comes into the frame. Right behind him, accompanying him, is a kelp greenling. He follows him through the frames. And sorry, here the blue areas are kelp greenlings kind of highlighted, and the red are Octopus Rubescence. You can see him following through the camera frames, and then as the octopus departs the camera frame, the kelp greenling follows him out of the frame. And we didn't just see this on one occasion, we saw this on several occasions. Here's another instance of it where we have an octopus coming into the frame, closely accompanied by a kelp greenling. They hang out for a while together in very close proximity. No aggressive interactions. It's kind of strange. And then the octopus departs, and the kelp greenling follows him. So now that we've examined these interspecific interactions, let's jump over to conspecific behaviors and their interactions. So how do these octopus interact with one another? So on the bottom of this is just like the other one, Pearson residual. So negative means that it's less than we expect, and blue and positive means it's more than we expect. So the octopus is in the frame. If we have more than one octopus, this can be in some cases up to 3 or 4, but often just 2, versus just one octopus in the frame. So if he's by himself, the odds of him periscoping are more than we would expect. So it's a higher percentage. And the fortification, it's not significant, but it's slightly higher than we would probably expect. Octopuses, however, in the frame, if there's 2 octopuses or more in the frame, they periscope significantly less. You don't really see any trend with the fortification, but it happens a little less frequently as well. So if I were to personify this, it seems kind of like the octopuses are hanging out in bottles. If there's more than one octopus, they don't have to keep as watchful of an eye, and so they rely less on periscoping, and they're in their bottles more. And this could just be they don't want to hang out with one another, so they're hiding in their bottles as well. There's a number of ways that you can kind of play this. Now let's jump over and look at how conspecific interaction affects social interactions. So how do these octopuses influence social interactions? If you have more than one octopus, so in this case, this would be more than one octopus present or absent. I'm sorry, this is just an octopus present versus absent. If an octopus encroaches into that area, so you have an octopus visiting the area, they'll come in, and more frequently, they'll interact with the bottle interior if an octopus is present. If you'd expect an octopus to be asocial, why would it be coming through a camera frame and reaching inside all the bottles more frequently when an octopus is there? Coupled with this, we never observed any den evictions, so they weren't like trying to drag each other out of their bottles. That's not why they were reaching inside. They were simply just passing through, reaching inside. Whether or not it didn't really matter to them, they were just doing it significantly more when an octopus is present. Now let's jump over and look at our conclusions. So we've overturned a lot of our previous understanding of the species. First, they're not nocturnal. Octopus rubescens is active during the day, and it fortifies during the night. And this is probably due to just biases from our observations being primarily in captivity. Second, kelp greenling like to hang out with octopus. If you talk about interspecifics, they spend more time and they come by more frequently when octopus are there. And they seem to hang out as they kind of move camera frames. Finally, octopus rubescens like to hang out with one another. They will spend more time at den locations, and they'll reach inside bottles more when another octopus is present. Also, no den evictions were observed. And this is kind of in stark contrast to a paper I didn't have time to go into, but basically in 1976, there was an individual who captured a bunch of octopus rubescens, put them in tanks together, and gave them like a finite amount of dens. So you have 3 octopus, 2 dens, 1 den. She watched how they interact. They observed a ton of kind of eviction behavior, where one octopus, a bigger octopus, will come in, drag a smaller octopus out of its den, and take over its den. This behavior, out of almost 70 interactions, she observed it happening about half the time. Every time an octopus would interact, about half the time of those, they would rip another octopus out of the den and inhabit its bottle. However, among all of the interactions we observed, we didn't see a single den eviction. And that was out of about 43 interactions. So significantly, like 0 compared to about half the time in a laboratory type setting. So this kind of indicates a level of behavioral plasticity that we see in this species that we didn't previously understand or know. So with that, thanks for your attention. I want to acknowledge all the help I've received on this project from my advisors, the Dive Associates, in addition to all the free gear from NOAA and Northwest Marine Technology. Especially, I want to thank my major professor, Dr. Kurt Onthank. He's the most talented, knowledgeable person I know, and somehow he manages to be so incredibly humble and forgiving of my mistakes, which are very plentiful. So he's given me the skills I need to succeed in graduate school, and he's really given me the confidence I need to continue on towards a PhD, hopefully. And I thank him for his friendship and his guidance. Finally, I want to thank my undergraduate research team for all the data analysis they did, all those late nights, and all the photos of animals zooming along the ocean floor. I know it gets tedious after about the third crab, so thank you. All right, so do we have any questions? Yeah, Rachel.
Did you see any predation events while you were?
We didn't see a single predation event. However, on multiple occasions, we'll see a large lingcod or a kelp greenling sitting about that far away from a bottle entrance that's about this big, and there's tentacles just hanging out outside the bottle, which in my experience, you would expect them to at least take a bite, but for some reason, they just sit there, and they're like entranced by it. Like they'll put their eye right up next to the bottle opening, and they'll just sit there, and they'll switch back and forth and look in at the octopus. They won't take a bite. It's the weirdest thing. And going into this, I anticipated all kinds of predation events. I thought sharks and lingcod and kelp greenling, all kinds of stuff would be taking them out of their bottles. But nothing. So it was very weird. Other questions?
When you were addressing den evictions, I'm just curious, the area that you were doing all the research in, there were a lot of bottles. And you referenced the other paper where, they put, 2 dens, or 2 bottles maybe in a tank, and there's 3 octopus, which is naturally,
Yeah.
So how would that influence the area you were in? Were bottles just plentiful? Do you think that would affect den evictions?
It could affect den evictions. It's strange. The paper I was referencing, Eleanor Dorsey, she went out from Friday Harbor, which is right there in Puget Sound. She put down a pretty large trawl, pulled it along the ocean bottom, and she would collect Octopus rubescens primarily in bottles or sometimes in little barnacle shells kind of hidden down in there. Out of 666 trawls, she got about 30 octopus, and we got like about 50 or so octopus in like a handful of dives. So this area, and she was trawling all across Puget Sound. If you would give each trawl, she pulled it about a meter. Out of 666 trawls, you'd expect her to find more than like 20 octopus. So our population density somehow appears to be way higher than you would find out there. There's also some difficulties with bottom trawling in that, like, when you're dragging along the bottom, octopus can get out of the way. They can slip through nets, things like that. So it's not terribly accurate, but it seems to indicate that this population is at a higher density than you would expect. So, Dr. Onthank?
Yeah, so 2 things. Number 1, trying to repeat the question after it's
Yes.
for those online. And then also I have a question from online. Someone asks, how many different species of octopus did you observe during your research?
How many species of octopus did we observe during our research? We found 2 different types of octopus in this area. There's much more in Puget Sound than that, but we found baby giant Pacific octopus inhabiting these bottles, stumps, boards, and a toilet, I believe. And then we found a bunch of Octopus Rubescence. The primary octopus in this area is Octopus Rubescence, but the giant Pacific babies can be found from time to time. It's a little bit more rare, though. So I found and tagged 4 of them and released them back to the site, but we never saw them again. And that's probably just because they're so large, they don't like to inhabit the bottle if they can find something better. There's a lot of debris down there that's bigger than a bottle that would allow them to inhabit those areas. Any other questions? Yes.
So you said you tagged around like 50 or so. Consistent throughout the time period, did you keep seeing the same ones, all 50, or did some disappear from observation?
Yes. So among the octopus that I tagged, how many were frequently like spotted a second time? We found some that were at the beginning of the deployment, and even though we collected 33 days worth, we found the same ones about 40 or 50 days later. Because we didn't get data every single day because the camera would die, tides are bad, we can't dive it, we have to wait a few days. So over the 45 days or so that they were all deployed and we were taking data actively, we saw octopus from the first deployments on some of the last deployments as well. So well over a month later, still kind of hanging out in that area. Unfortunately, though, we encountered about, I think, 30 or so octopus that were tagged, and some of those were the same octopus several times, compared to the 50 or so that we tagged. So they seem to kind of disperse and go their separate ways. We all collected them in that area and put them right back in that area. So I had anticipated they would stay there a little bit longer. We'd see them hopefully a little bit more, but they seem to kind of be transient, moving on. And that could be because there's bottles up and down the beach, so they can inhabit any number of areas because bottles aren't a limiting factor in their movement anymore. Any other questions? Dr. Cowles. Yes. Yeah. So if an octopus just zooms through the frame, it's not of any big consequence because we don't really get a lot of data from that anyway. So it's ideally nice to know that it's like tagged and which octopus it is and how it's moving. But we aren't doing like spatial, like how far are they moving and stuff like that. So if they just pass through, odds of you getting a tag is pretty low. Because you might get 2 or 3 photos and then they're gone, and they're usually teetering along the bottom. They're all like camouflage. All their papillae are erected. So all of those like those fluorescent tags are kind of deep under those like structures, darkly colored skin, difficult to see. However, yeah, during the day it makes it a little bit more difficult as well. However, if the octopus are there for any extended duration and you get them a few comings and goings, even just poking their kind of nose out, if you're able to saturate it, so like take the photo, blow the saturation way out, you can see like blue, yellow, red like lines, and it's actually not crazy difficult to distinguish that because I didn't edit these photos because they look kind of weird. It's not as obvious, but all you really need is just like a little tiny bit of that tag, and you know which order it's in, and because each one has a different order, you're able to distinguish it. So, good question. Anyone else? Shane? Yeah, I might. I'll move that.
So with the fluorescent tagging, have you thought at all about how you might standardize or scale your like tagging system? Because it seems like it would work well for it's like an easy, non-invasive way to tag octopus, and octopus is a pretty important commercial species. There's a lot of tagging programs that have a lot of trouble. And
Yeah.
just using like, can you draw letters or numbers? Like, have you thought at all about how you might identify them in a way that can be like logged into like some kind of national like database or something like that?
Yeah. So the question was, is there a way to standardize this tagging technique for use with wildlife dynamics, tagging lots of octopus, watching their movements? Absolutely, Shane. This could be used for a lot of that application. Unfortunately, nobody's really applying it to cephalopods. It's been mostly used for kind of fishery type lizards and newts using them, because you can just put like a little tag on its leg, just a little dot of pigment, you know which one it is. So there has been some application for that, but I'm not sure if the octopus fish is big enough for them to be like, oh yeah, let's tag a bunch of these. Additionally, some problems that I foresee might be putting basically inside the tissue of octopus. Imagine eating some calamari and you're like, wow, that is very cheap. Octopus, it's, you know, some silicone you're ingesting that's fluorescent. So I could see that it has a lot of application. If you could find an area that you could tag that wouldn't be eaten, I think it would have a little bit more application. But unfortunately, I think people eat most of the octopus, so that might raise some difficulty.
Sorry, didn't you say they used them for salmon too?
Yeah. Well, it's tagged in the fin. So unless you're eating the fin with fin soup or something like that, yeah. And it's used for like salmon fry, and I think it's on the adipose fin, which is possibly clipped, or like the back of the top fin. So anyway, but yeah, no, it absolutely has application. I just don't know if anyone's applied it yet, or if anybody cares. Because like honestly, the amount you make every year in the salmon industry is like octopus pale in comparison to that. So I don't think anybody's like, oh yeah, let's take the time and the money to try and track octopus. Although I think it's cooler than salmon. I don't know if it'll have much application for a while yet, but yeah. Yeah.
Okay, so question from online from Lloyd Trubot. Question: do octopuses like to take a midday nap? Any suggestions why there seems to be no activity between 10 a.m. and 2?
Yeah. So let's jump back and look at that. Oh, okay. So do octopus like to take a midday nap? Why are they not very active between 10 and 2? I don't know. My kind of anticipation is early in the morning, they're out and about, they're foraging, they're looking for things, they're departing their dens, and they're moving through the frame a lot. And then through the middle of the day, they're probably back in their bottles or they're out foraging. And then towards the evening, they kind of come back. You start seeing them a lot more frequently under the cameras. They're starting to occupy those bottles more as they're kind of bedding down, if you will, for the night. And then we get hit with this big fortification event. Nighttime's coming, time to close the door. So if I were to personify the whole thing, that's how I'd anticipate. They're either out foraging during this period or they're kind of holed up in their bottles because they're not hungry. So, any other questions?
I want to jump back to your camera system. I know there's a lot of problem solving with that. I was wondering if you could just share a couple of your challenges and discoveries that you made with that, and maybe address the lenses.
Yeah. So challenges and issues that I encountered with the camera system. So unbeknownst to me, this is very common apparently among people who do this sort of thing a lot. UV won't go through plexiglass. I did not know this. So I was trying to bump up the voltage on these to like insane amounts. And I was trying to limit like my battery capacity. How long can I run it? But also just like have a massive light pulsing UV out. Turns out, plexiglass, the stuff that's like commercially available, it stops about 99% of UV. So yeah, I could see it. It's like blue in there, but no UV was passing out. Could not figure it out. OnThing told me like, hey man, I think this is happening. I was like, no, that couldn't be it. And I continued on for like another week thinking that was impossible. So it ended up, I had to cut glass to fit over the UV. So the UV is able to come out of the glass, and then when it goes back into the camera system, all that extra background noise, all that extra UV is actually canceled out by this plexiglass. So it makes for a pretty slick system in that the wavelengths that are reflected back here are what are fluorescing off the octopus, which isn't UV anymore. It's what's basically fluorescing back. And that's actually able to pass through this and be accepted by the camera, whereas the UV drowns out. So you don't see any of that bluish tinge, which is nice because it acts as a camera filter as well. So that was one issue. Another issue that I encountered is somebody went out and cut one of my buoys while I had a camera system down doing some tests. And so I thought it had floated away and it was out in the strait or just gone. And things around Puget Sound can drift away quite quickly. So we went out with, I believe it was Phoebe Cherney, and we went out and we dropped a weight where we thought the camera had been. We dove down in about 5 minutes. We found the camera again. And that was really lucky because that was about $1,000 plus of camera equipment. And I didn't have enough money to build a whole nother system or enough time. So that was really nice. Yeah, good question. Anybody else? Dr. Arnthing.
Another one from online. Were you able to compare wild behaviors to captive behaviors and make any correlations or assumptions?
Okay, the question was, can I compare wild and captive octopus behaviors and make any correlations? So I made a bunch of correlations, and I ran kind of some numbers on my own to look at trends, but this is a paper from like 1976 that was never published or really peer-reviewed to any major extent, and it was based on a person who just put a bunch of octopus in a tank and just sat there and watched them. So the comparisons between the methods are pretty stark. So I would love to make more in-depth comparisons, but I'd have to go out and basically visualize these octopus wild, then try and capture the same octopus, bring them into a lab, and see if their behavior changes. So I can make a lot of assumptions about it, and I have made some in this presentation, probably more than I should. But if I had more time, I would actually do a study where I go out and capture octopus, the same octopus that I'm surveying here, and see if their behavior changes. And that would be a clear indication of behavioral plasticity and kind of social adaptation in these species. Dr. Cowles. Yes.
Is there a reason why you were not able to record the status? Are they in the bottle? Are they in the area? Out of the bottle, but in the area, and they're out of the area. So you can't, if you can't see them, can you tell whether they're gone or whether they're in the bottle? Or you can't see them.
Yeah. So the cameras are really, really good. So I saw it every time it came and went. Oh, sorry. The question was, if an octopus is in its bottle, do I know if it's there versus somewhere else? And how do I use frame departure, not frame arrivals? Is that kind of it? So a portion of why I use frame departures as opposed to frame arrivals is I don't care what an octopus is doing in that frame. If an octopus is out roaming around or foraging, say, the best indication of that is the fact that it's no longer under my camera. So the second it leaves as a frame departure, that's when I say it's out. That's just marked as it's. I'm assuming it's no longer under my frame. It's out somewhere else doing some other type of behavior. So that's just marked an activity period. If I were to indicate every time an octopus showed up, it might show up, go inside a bottle, and then sit there for an hour and not actually do any kind of a foraging or scavenging type behavior. So it is an assumption based on the fact that they're departing. I'm assuming, hopefully, they're out foraging around. At least they're moving to another bottle. Whereas if they're entering the frame, I'm not confident that they're actually doing any type of behavior. And then remind me the second part of that. It was the whether or not they're in a bottle.
Right. If they disappear from sight, do you know if they're in the bottle or not? I mean, that's quite different.
Yes. So frame departure means they left the actual frame, but I have a whole nother behavior category in my in this, which basically like I have an enter and exit of the bottle, and I have a whole nother figure for that, but I didn't show it just for the sake of time and explaining the difference between an enter and an exit of a bottle versus a frame departure. But a frame departure means they're not anywhere near the bottle. They're actually leaving the camera frame, what I can see, whereas every time they entered and exited, I was able to capture that. And I... Yes. So the reason for that is I, through all this data analysis, out of all these interactions, I never once encountered an octopus that came out of a bottle without previously going into the bottle. So there's never an instance where I saw one leave that I hadn't already seen it come back. If I did see that, I could jump over to the second by second frame, and we actually did go through a lot of the second by second frame. I actually went through all of it surrounding every octopus interaction, and I never found an instance where an octopus left and then all of a sudden it leaves again, because that would indicate it came back and I didn't see it. I also never had any type of interaction like that where something left the frame without first coming into the frame. So no fish, no crabs, nothing else. It was able to capture every single interaction. So, any other questions? Those are very good. Dr. Onthick.
One more from online. It says, what programs and techniques were used in designing the camera system?
Okay, what programs and techniques were used in designing the camera system? That is a very, very in-depth question. So Raspberry Pi 3B Plus is the base foundation of that. And you can run different, basically, computer code on this. Imagine it like kind of a little tiny computer. I can hook up a HDMI cable to it right here, and I can attach a screen to this and a mouse and actually go through this like it's a desktop. So the code that's in here runs all of these systems. So this is called a GPIO pin, General Input Output pin. Basically what it does is it runs little electrical wires through this PCB, this circuit board, and it's able to control these different components. So this is a clock here. This is a Pro Trinket, which you can code with its own kind of algorithms in order to run the strobing system. These are current and voltage regulators. This is some on-off switches for the voltage regulation. Then you got some resistors in here and fuses. And then this front component is basically an LCD screen that allows you to see if the program's running, is it working correctly, because this Raspberry Pi gives printouts to this. So imagine these all as accessories to the real kind of workhorse here, which is a Raspberry Pi 3B Plus, which is consumer-grade electronics. So I can get online, and it used to be for about $50 I could get one. Now, because of stuff, it's about $200 or $300 to get one of those. But yeah, it's basically just a very simple computer. Did that answer the question that they had? Okay, perfect. Any other questions?
Okay, well, thank you very much, Jesse.
Thank you.